Okay, so ladies and gentlemen, you are welcome. So in today's video, we are going to look at the chemical properties of an amino acid. So amino acid undergoes a series of reactions which include decarboxylation, esterification, and so many other types of reaction. So now the first types of reaction that we are going to look at is decarboxylation reaction. So when you say decarboxylation means is the removal of CO2. So decarboxylation is the process whereby CO2, that is carbon dioxide, is removed from a compound. So amino acid undergoes the carboxylation reaction to produce corresponding amines. So the amines, thus amines are produced. So like for example, we have histidine. So histidine is an amino acid which actually decarboxylated to produce CO2 and histamine. So histamine is an amine derivatives that derive from histidine as an amino acid. And then we have tyrosine. Tyrosine also is decarboxylated to produce tyramine, and lysine also decarboxylated to produce uh, cadaverine. So what are these important amines that are generated from the carboxylation of histidine, tyrosine, and lysine? So, so this correspondence amines generated from the amino acids histamine is, of course, a neurotransmitter that aids in cellular communications. So they are found in the brain and the spinal cord. Then we have tyramine and cadaverine. They are all also amino acid and neurotransmitters that are generated from tyrosine and lysine. In fact, cadaverine is also can it's serve as also hypotension compound. It can be used as the drugs in disease treatment. So then the next types of reaction apart from the carboxylation reaction is a reaction with anhydride. So amino acids are known to actually react with anhydride. So you see this is one, we have alpha amino acid here where the anhydride react with the amino acid to produce aldehydes, carbon dioxide, ammonia, and hydrogen. So this is actually what is actually generated after the reaction. So you see, ladies and gentlemen, if you can see clearly in these types of reactions, you can look at what we have in the reactant and also what we have in the product of the reaction. So you see here initially before the reaction takes place, you see here we have OH and then here we have OH. What is the difference between the product and the reaction? So in hydrogen and hydrogen is that if you look at it after the reaction, O is removed. So oxygen is removed, and therefore also aldehyde is generated as a byproduct with CO2 and ammonia. So this is in hydrogen, reaction of the amino acid with hydrogen. Yeah, the amino acid after the reactions generates four major products. So ladies and gentlemen, the most important thing here is when we talk about the reactions of amino acid with hydrogen, the first thing that will come into your mind is the product that are generated. So four major products are generated, then hydrogen, and then aldehydes, carbon dioxide, and ammonia. They are the major products that are generated. So this is actually the second types of reactions of amino acid. Then the next types of reaction is the reaction with a reaction of the amino acid with alkalis, that is salt formation. So you see, generally amino acids react, you see carboxylic group of amino acid can release hydrogen ion with the formation of carboxylate ion. So we can see clearly here we have sodium hydroxide. So sodium hydroxide here react with the hydrogen from the carboxylic group to generate water molecules and then corresponding salt, which is sodium carboxylate. So we have sodium carboxylate, which is actually a salt of an amino acid. So also at the same time, apart from the reaction with alkalis, amino acid also react with alcohols to generate ester. So you see the first types of reaction, we talk about the decarboxylation reaction of an amino acid, and then second, we also talk about the reaction of amino acid with alkali, reaction of amino acid with uh, hydrogen, and also now the reaction of amino acid with alcohol. So you see alcohol like here, we have alcohol, which is ethanol. So you have ethanol react with the amino acid to generate, it actually react with the amino acid here to generate ethyl ester of the amino acid in the presence of the acid catalyst that promote or facilitate the reaction. So after the reaction, the ethyl ester of the amino acid are generated plus water molecules. So this is the esterification reaction of an amino acid. So then the next type of reaction is actually the reaction of an amino acid with densyl chloride. So here, if you can see clearly, a densyl chloride means the D stands for dimethyl, A stands for the amino, naphtha sulfonyl chloride. So this is actually densyl chloride. So the densyl stand for dimethyl amino naphtha sulfonyl chloride. So the compound, which is the densyl chloride, when we act with the amino acid, is actually generate a compound which is a fluorescent densyl derivatives. So see here we have the densyl chloride here react with the amino acid, and the products generated is the densyl derivatives of the amino acid, which is a fluorophore. A fluorophore. So it's used as a fluorophore, it's used as a dye in fluorescent or in fluorometry. So then another types of reaction is actually the reaction of the amino acid with with acylating agent. So acylating agent here is actually react with the amino acid. So you see, when amino acid react with acid chlorides and acid anhydride in alkaline medium, it gives the loyal amino acid. So you see, we also have the acylation reaction of amino acid. So amino acid react with acid chloride and acid anhydrides in an alkaline medium to generate colloidal amino acids. So this is actually, ladies and gentlemen, the different types of reaction of an amino acid. So then the last one is actually, so the other types or the next types of reaction is reaction of an amino acid with Sanger's reagent, especially in the sequencing. So the Sanger reagent is 1,4,2,4-dinitrobenzene, which is called a Sanger reagent, FDMB. So the word FDMB stands for Sanger's reagent, which is an acronym of 1,4,2,4-dinitrobenzene. So this is actually the name of the Sanger's reagent. So this Sanger's reagent is known to react with an amino acid to generate a yellow compound and then hydrogen fluoride. So you see, the Sanger reagent react with the amino acid to generate dinitrobenzene amino acid, which is yellow in color. You see, the major product here is dinitrobenzene amino acids. So then the next thing is actually the reaction of an amino acid with Edemann's reagent. So Edemann's reagent is standing as phenyl isothiocyanate. So this phenyl isothiocyanate react with amino acids react with the amino acids to give phenyl thiodantoic acid. So finally, it turns into cyclorized form, which is known as phenyl thiohydrantoin, which is an Edemann's derivative. So this Edemann's reagent is a reagent that also used in determination of an amino acid sequence. Both Sanger's reagent and Edman's reagent, they are reagents that are used in determination of an amino acid sequence. So, ladies and gentlemen, this is actually brief types of reactions that take place between amino acid and other reagents. So, this is the reaction of an amino acid. So, please don't forget, if this is the first time you come to my YouTube channel and join my YouTube community. So, you see, we can see clearly here how the reaction takes place. So, bye bye, ladies and gentlemen. See you in the next video.